but today's lab is on water quality analysis. And so when it comes to water quality, we know we can test for all different kinds of things in water, right? Things like lead, but we're not focused on chemical contamination in this course. We're focused on microbiological contamination. So, do you expect to find bacteria in water?
Yeah, probably.
Sure, because remember, at the very beginning of lab, when we did the environmental plates, we learned that bacteria are everywhere, right? So of course they're going to be in water, and they're going to be in your drinking water. So is that a cause for concern? No, right? Because we already know, right? Bacteria are everywhere. You inhale them, you eat them, you drink them, right? They're all over. So we're not worried about just bacteria in general. But when it comes to drinking water, our water does come from the environment, right? It may come from groundwater if you have a well, or rivers, streams, and reservoirs, but they all contain bacteria. And when it comes to our water supply for this area, where do we get our drinking water? Where does our drinking water come from? What's our source of water around here?
Is it spring?
Mm-mm. Where do we get our drinking water?
Groundwater.
Nope. At the eastern half of Maryland, Northern Virginia, where do we get our water?
The streams.
More than just a stream.
Reservoir.
I didn't hear that. Where?
The reservoir.
No. You guys don't know where your water comes from? Bryant, do you know? Craig? Roxy? Christina? What river is next to us?
Does it come from Baltimore?
Huh?
In Baltimore?
No, that's Baltimore, and that's Western Maryland. What's near where we are?
The Potomac River?
The Potomac River, right? The Potomac River. Have you guys ever been to the Potomac River? You ever been to Great Falls?
Yes.
Yeah. You guys need to go check it out. It's really beautiful in many places. But our source of water is the Potomac River, which is not necessarily the cleanest river, but of course they treat the water before it's delivered to your tap. Now, when it comes to bacteria, of course there's lots of bacteria in the water. We're not concerned about all bacteria, but we all are concerned about bacteria called coliforms. And as the name coli implies, these are bacteria that live in the intestines of animals. So coliforms are gram-negative rods. Of course, they're not spore formers, and they do ferment lactose, which helps us to identify and differentiate them. But when it comes to coliforms like E. coli and its friends, isn't your gut full of E. coli and its friends? All the intestinal bacteria you have, how did they get there?
From the outside, somewhere.
But how did they get inside of you?
We eat them or drink them.
Right. You ate them, and you're still eating many of them today. So if we have coliforms in the water, why does that concern us? Right? Because there's coliforms inside of us. We got them by eating them from the environment. So why are we worried about coliforms in the water? Because if you find them, that suggests fecal contamination, right? Feces in the water. But most of those normal fecal bacteria are part of our normal flora, right? So again, why are we so concerned about it? The reason why is that there are potential pathogens associated with feces, like salmonella and shigella, even viruses like hepatitis. So there is the potential for pathogens to be there if fecal contamination is a problem. And so we use fecal coliforms as indicators of water quality. So if we find fecal bacteria in the water, that's not a good thing because there could be potential pathogens there. Now, one of the things about pathogens, let's say that you do have, let's say you live on a farm. So you have a well, right? Because you're too far away to be hooked up to water and sewer lines, so you have a well. So then when you flush your toilet, where does it go? Well, what they do is they basically bury a humongous tank in the ground. And when you flush your toilet, everything goes into the tank, and the tank has holes in it. And so bacteria do help to degrade waste that's in the water, and then it leaches into the surrounding soil. And so you're not supposed to have your septic tank near your well, right? Because the seepage could contaminate the groundwater, which sometimes happens. So if there's someone in your household who's sick, let's say they have food poisoning, so they have salmonella, they'll be releasing that in the feces, it gets flushed into the septic tank, and it drains into the soil and ultimately leaches into your groundwater. But once that person recovers, there's no more salmonella. Right? Salmonella does not persist in the environment. So if you go to check the water a few weeks or a month later, you will not find any salmonella, right? Because the person recovered, so no more salmonella being released. But if fecal contamination is an ongoing problem, you will find fecal coliforms. So we use those as indicator organisms, right, to indicate water quality. And it's fairly easy to isolate and selectively grow and differentiate fecal coliforms like E. coli. So there are several different ways to do it. We're going to talk about one technique. So we use the fecal coliforms like E. coli and its friends as indicators of water quality. If we find them, that suggests feces is contaminating the water supply. And we don't want that because there could be pathogens associated with feces. When it comes to the U.S. Public Health Service standard for water quality, drinking water should have no more than 1 coliform per 100 milliliters. So no more than 1 coliform per 100 milliliters. When it comes to all other bacteria, no more than 100 per 1 milliliter. So it's okay to have bacteria in your water because they're always there. There's just certain kinds we do not want to find, like fecal coliforms, right? Because that could suggest your water is not safe to drink. So we're going to test, or we're going to talk about how to test different water samples using a membrane filter. So we're actually going to filter a water sample through a membrane that traps the bacteria in the membrane. All the water, even viruses, those can pass through the membrane, but the bacteria will get stuck on the membrane. And then we will use a nutrient medium to grow those bacteria to form colonies. And the colonies will change color, and the color will help us to differentiate what type of coliforms they are. Now, there are some advantages and disadvantages to using a membrane filter technique. Advantages: it's relatively fast. In 24 hours, you get colonies, so you can see your results. It's sensitive. You can filter more than 100 milliliters of water in order to find, right, those few bacteria that might be there. So it is sensitive. And it's quantitative, right? It's accurate because you get colonies that you can count. And you can do a simple calculation and determine the number of coliforms per 100 milliliters of your sample. So it's fast, accurate, quantitative, sensitive. So it's a good technique, but it also has some disadvantages. Your water sample does have to be relatively clear. If it's muddy, if it's really turbid, then your filter will get clogged. When it comes to how many bacteria are actually in the water sample, if your sample is really highly contaminated, your Petri plate will have solid bacterial growth, and you won't be able to see any individual colonies. So that would require you to perform serial dilutions and repeat the membrane filtration multiple times. And so this stuff does cost money, right? So that would be a disadvantage. So your water sample has to be relatively clear and relatively uncontaminated. So not too many bacteria in the sample, right, that you test. So we use the M. coli blue system, which actually turns E. coli colonies blue. So it's really easy to differentiate E. coli from the other coliforms. And the other coliform colonies will appear red. Now, the old system we used to use used endo broth, and endo broth is similar to EMB, right, the eosine methylene blue. And as you recall, E. coli is shiny green on EMB. And so the endo broth, we would look for shiny green or shiny gold colonies. The problem with the endo is that it did not work as well as the M. coli blue. M. coli blue is crystal clear. It's blue, and it's E. coli. So it's much easier to use and much easier to differentiate E. coli. So that's why we use the M. coli blue. So what we would do in class is at each table, there would be two groups. One group would test pond water from the pond that we have on campus, and the other group would test aquarium water from one of the fish tanks that we have in the department. So half the class ends up doing pond and the other half ends up doing the aquarium water. Now, because we do not know how contaminated those water supplies are, we use a 1 milliliter sample. So it's just sort of a general standard to use 1 milliliter. It will make the final calculation easier to do. But if you suspected you had really highly contaminated water, maybe you only use half a mil or 0.1 ml instead. If you think your water is pretty clean, maybe you use more than 1 ml. But 1 ml is sort of a standard amount because we don't really know how contaminated our 2 water samples will be. Now, you cannot put 1 milliliter onto the membrane filter because it would just, make the filter wet and that's it. So you do have to suspend your 1 milliliter sample into 100 milliliters of sterile water. So that way you can filter the 100 milliliters. And so you have a nice even suspension, right, to filter out and separate the bacteria as the water goes through the filter. So once you have your sample ready to go, you load it into the membrane filter apparatus. And so there's actually a plastic cup system which has a lid and a bottom, and then the middle part is like a clear plastic cup. And the lid and the bottom will separate from the cup so you can create a cute little petri dish using the bottom and the lid. But you would pour your sample into the membrane filter apparatus. It's attached to a vacuum manifold, so we're going to create a vacuum, right? We have a pump. It will suck the water down through the membrane filter, filtering it, and then we collect the water in a flask. But we all filter our samples, suck the water down through the membrane filter and trap the bacteria. And then you separate the top and the bottom from the middle part and put the top and the bottom together to make a cute little petri dish. Then you add the M. coli blue broth right to the membrane. So we're providing some nutrient medium or some nutrient broth. You do have to put a plastic cap on the bottom because remember the bottom portion is open so we could suck the liquid through, so then you just have to plug it with a cap. But you have these cute little petri dishes. And then you add your M. coli blue broth, you incubate, and the next class we observe the results. So here we see an example of one of those plates. So you can clearly see several larger blue colonies, right? So those blue colonies are what?
E. coli?
E. coli. We also have some smaller red colonies, and those are other coliforms, not E. coli, but other coliforms that are there, so we count them as well. If there are any other colonies that are any other color besides blue and red, they don't count, right? There's some other kinds of bacteria, they're not coliforms. So we're just concerned with red and blue colonies. Now, ideally, the number of colonies should be between 20 and 80, but it depends on your water sample. 80 is sort of a maximum because at that point on these smaller plates, because these plates are only about 2 inches in diameter, so they're pretty small. And so if you have more than 80 colonies, they're all growing into each other, so it's hard to see them as separate. It's analogous to the 300 colonies on a regular agar plate, more than 300, right? They all start to merge together so you can't see them as separate. So it's the same idea here, but we would always count whatever we got on the plate, right? Because our samples tend not to be that contaminated, so we just count whatever we have. So after you count your red and blue colonies, you will divide by the sample size, which was 1 milliliter, so that's easy. and then multiply by 100. And that gives you the number of coliforms per 100 milliliters in your sample. So I've given you some sample data. Like I said, normally we would have 6 sets of data, but I'm just simplifying it here so we have some numbers to play with. So from the pond sample, we counted 11 blue colonies, 18 red colonies for a total of 29 coliform colonies. In the aquarium sample, 2 blue, 7 red for a total of 9. Right, which is good. We hope that the aquarium is cleaner because we clean it out periodically, right? So we do the calculations. So no complex math here. So what would be the coliforms per 100 mil in the pond sample? No calculator required for this. Multiply 29 times 100. And what do you get?
290.
Right, so 2,900, right? Or 2,900. And for the aquarium, what's 9 times 100?
900.
900, right? So the calculations on this are easy. Now, there is a fancy word for drinkable, and that fancy word is potable. You ever heard of potable? It's a fancy word for drinkable. So based on these results, is your water sample potable? Is it drinkable? So would you drink the pond water?
No.
Would you drink the aquarium water?
No.
No, right? Because what was the water quality standard? What was the water quality standard for coliforms in water? What's the standard for potable water?
1 coliform per 100 milliliters.
Right, so no more than 1 coliform per 100 ml. So I think we've exceeded that, right? 2,900, 900, right? It's a lot more than 1 coliform per 100 ml.
I have a question. How can we include the red in the total if that's not a coliform?
Red are coliforms.
Oh,
So
and
blue is E. coli specifically.
oh, got
But
it.
red are other coliforms that are not E. coli. So things like Enterobacter aerogenes or Klebsiella pneumoniae. So there are other coliforms present, right? Just not E. coli.
Got it.
So what are some things that you can do to make this water potable? What could you do to make it drinkable?
Maybe boil it.
You could boil it, right? But what if there's no power? What if there's a natural disaster and you can't make heat, right? So no fire, no electricity, no gas, so no way to heat up the water to boil it. What could you do to make it safe? So in a natural disaster, what could you do to make your water safe to drink? What, no ideas?
Can you boil it?
Again, there's no way to make heat. So how could you make your water safe to drink? Does everybody have microbiological filters so you could filter it? Now, most people don't have those, right? But most people do have something in their homes they can use to make the water potable. What could they use to make the water potable? Something almost everyone has in their homes they could use, right, to make the water potable. What is that? I didn't hear that. What was that?
I was saying, could you use salt?
Salt?
Yeah.
You need an awful lot of salt, right? More than 7.5%. So that would be an awful lot of salt. And then you wouldn't be able to drink it because it's way saltier than the ocean, right? So that wouldn't really work. Something almost every household has. Having to do with laundry.
The bleach?
Bleach. Bleach. So you can use bleach to disinfect water to make it drinkable. In fact, it's been a long time, but bottles of bleach used to have instructions on the bottle, right, to tell you how to disinfect your water using bleach in an emergency. But that's just plain bleach, not these bleaches with perfumes in them, or the bleaches that have thickening agents so they don't splash when you pour. But just plain bleach can actually be used to disinfect your water. Although they stopped putting those instructions on a long time ago, probably because they were afraid that stupid people would drink the bleach, right? Thinking, oh, I have a stomach infection. I'll just drink bleach and I'll take care of it, right? Well, that will take care of it in you.
But how would
You
you
can,
drink the water?
huh?
So you would pour the bleach in the water and then just drink it?
Yeah. But you add about a third of a cup of bleach to a gallon of water, or 1 or 2 tablespoons per quart. And then you mix it, and you have to leave it, for a while. And then you can agitate it, like pour it back and forth between containers to aerate it, and that helps the chlorine to evaporate off so the water is safe to drink. But you can't use bleach because the water that comes out of the tap is chlorinated water, right? So it's chlorinated water. And a lot of people don't like the taste of chlorine, so they use carbon or charcoal filters to remove the chlorine. Right, so it just tastes like pure water. But you can use bleach to disinfect your water in an emergency. So you should always keep some bleach on hand because it's useful. And like I said, they used to put the instructions on the bottle, but have not done so for a long time now, right? Because they're afraid of liability. But you can use bleach to disinfect water. If you've gone into swimming pools, how do they disinfect swimming pools? They use chlorine, right? So We use chlorine a lot. You could also use iodine. If you've ever camped, you know that sometimes you can't bring enough water with you, so you're going to use water from the local stream. But of course, you can't drink it right out of the stream. That would not be safe. So they do make iodine tablets to add to the water, right? Because iodine is a very good disinfectant as well. In fact, it's better than chlorine because iodine is more stable, right, than chlorine is. So you could use iodine tablets. The water does not taste very good because iodine also does not taste very good. But if you've ever been to the C&O Canal towpath, and it sounds like most of you have never been to the C&O Canal, but if you bike or hike or camp along the towpath, they do have water stations in the summertime. So you actually have a hand pump, and you pump the water to get it out from the ground, and they use iodine to treat the water because iodine is much more stable than chlorine. And the water, the groundwater, is right next to the Potomac, so they do need to disinfect it. And it doesn't taste very good, but it is disinfected, so it's safe to drink. And if you need water, then you can get water at various stations that are set up along the towpath. If you use that water to cook, like potatoes or rice, it'll make the potatoes and rice turn a darker color because the iodine is reacting with the starch in the potatoes and rice, right? It's like a starch test. We add iodine and it turns dark. So it is kind of weird to see that, but it's perfectly safe. But you can use iodine to disinfect water. Or many hikers and campers bring little special filters, and they actually can filter the stream water and remove bacteria and other parasites so they can drink it. So, how do you recognize coliforms on the M. coli blue Petri plates and E. coli specifically? Right, so here's the plate. So how do you recognize coliforms?
With the colonies, and then the blue ones are the E. coli specifically.
Right, so coliforms are red colonies, and E. coli are blue, right? And that's how you can tell. So why are we concerned about coliforms in drinking water?
Because it might indicate fecal contamination.
Right. Which may include pathogens like, Salmonella or Shigella, or even things like hepatitis, right? So we don't want feces contaminating the water because it may contain pathogens. Why are coliforms used to indicate water quality?
Is it because they're also gram-negative rods? Or they're from the infected?
Well, in addition to indicating fecal contamination, right, they're easy to grow and differentiate, right? So we can tell, usually using lactose fermentation, right? So we know that fecal bacteria, coliforms, are indicators of fecal contamination. They're easy to grow and they're easy to differentiate. And lactose fermentation is a common way to differentiate them. So describe the advantages and disadvantages of using a membrane filter.
Advantages could be that it's fast results.
Fast, accurate, right? Quantitative, sensitive, right? So lots of good things, but what are some of the disadvantages?
It costs money to keep doing it
Well, why would you need to keep doing it?
because it doesn't allow so much water through at once.
But why not? I mean, this is what you're trying to say, right? Right. If your sample's really turbid, really cloudy, really muddy, it'll clog the filter.
We can't see the rest of the document for some reason. It's like stuck.
Oh, really?
Yeah.
What are you seeing right now?
You just see like part of the answer to the first question and nothing else. I'm saying none of the other answers, just the top of the sheet with the plate and the charts.
Oh, really? I don't know why that would be. Let's see.
Oh yeah, now we could see the rest.
And I, yeah. Always technical glitches. So disadvantages, turbid water clogs the filter. Highly contaminated water needs to be diluted in a process of repeated multiple times. As we said, that takes time and money to do that. So there are some disadvantages. The advantages really do outweigh the disadvantages, so it's an excellent system to use to test water quality. And so how would you describe coliform bacteria? And so thinking back to the lecture on bacterial groups, we talked about enterics. What do we say about the enterics? Right, we learned characteristics of the enterics. So what are those characteristics? What's their gram reaction and shape? Gram reaction. This is an easy one. The gram reaction. Are you still there? Where are they? Gram what? I can't see that. You got to speak. What are they? I can't speak. I can't see chats. And what? Rachel, speak to me. You guys can still hear me, right? You guys say. Also, audio problems. Can you guys hear me now? Anybody out there? So can you guys hear me? Uriel, can you hear me? All right, well, I'll have to finish this up. Oh, we got disconnected, so the gram-negative rods. The intestines, ferment lactose, at least most of them, motile or not motile. They produce bacteriosis, right, which kills other great bacteria. So, unfortunately, we got disconnected. So, some of the things are trying to reconnect. But so I'm waiting while students are trying to reconnect, but it's not working. They're not able to join the session.